Okay, great. So let's get started. We'll let people slowly trickle in as we begin. Hi everyone, my name is Daniel. I'm the chair of CURBx and welcome to the 7th annual CURBx conference. It's a pleasure to be here and I can't wait to get this conference started. So first and foremost, I'd like to extend a big thank you to all our speakers for taking the time out of their busy schedules to present their research. We have an amazing lineup with a diverse range of topics and we can't wait for everyone to hear them. Before we get started, I'd like to go over some logistics. So if at any time anyone has a question for one of the panelists, please feel free to message me, Neha, or Tara, or just put them into the Q&A box, which is on the bottom tab for you guys. And we will answer them during the Q&A session for each panelist. And with that, let's get started.
All right, thank you, Daniel. Hi, everyone. My name's Tara, and I'm a member of the CURBx committee. To get started, I would love to introduce our first speaker, Erin Fleck. She is a junior majoring in engineering physics, and she is working on Fourier analysis of atomic resolution images. I will now hand it over to Erin to present her work.
Hi, everyone. My name is Erin Fleck. I'm a junior studying engineering physics, and I'm an undergraduate researcher in the Circuitous electron microscopy group. Today, I'm going to talk to you all about a Python platform that I developed that enables us to interact with materials at the atomic scale. Engineering, as we know it, is full of big ideas. Reaching 5G, which allows us to connect with anyone in the world at speeds like never before, achieving 100% renewable energy, or revolutionizing our transport system with trains that can actually levitate. However, to understand what makes these big ideas possible, we have to look much, much smaller at the atomic scale. It is often thanks to perovskites, a kind of repeating crystalline structure, that we are able to do or aim to do all of these great things. The exotic properties of various perovskites can often be exploited in order to build these kinds of systems. For instance, some classes of perovskites are high permittivity, low loss microwave dielectrics, meaning they're very polarizable and lose little energy during heating, which have made them suitable for all kinds of mobile communications that we use every day, like GPS, Wi-Fi, and radar, and has been crucial in developing 5G networks. Properties of other perovskites, such as high charge carrier mobility and strong optical absorption, have enabled perovskite solar cells to grow remarkably in efficiency in recent years, from 4% in 2009 to 24% in 2018. One of the most remarkable properties of some perovskites is superconductivity, which can be exploited to make superconducting magnets. These magnets have enabled the creation of ultra-fast, high-speed railways through the use of magnetic levitation or maglev technology. But despite all of these vastly different properties among different kinds of perovskites, they all share incredibly similar chemical structures. The difference between the next most exciting superconductor and, well, a rock, is fundamentally just one extra atom in the chemical structure. With the differences in these materials only being clear at the atomic scale, we obviously need a more powerful tool than just the human eye to analyze them. That is where scanning transmission electron microscopy, or STEM, comes in. STEM is an incredibly powerful tool that enables us to look at all kinds of materials at the atomic scale. Unlike regular optical microscopes, electron microscopes are not limited by the wavelength of light, which enables us to image a wide range of materials with an improvement of more than 4 orders of magnitude in resolution. In these microscopes, a high-energy electron beam is produced and sent through a series of around 100 lenses to precisely focus the beam on the sample. When this beam encounters the sample, it is deflected, and we can use the amount this beam was deflected by to determine how heavy a region of a sample is, and thus can generate an image in which brightness corresponds to heaviness, as we can see here on the right. Looking closer, we can see exactly how each little dot is actually an individual atom. With the aid of STEM imaging, we're able to see very clearly that many layered materials grown in a lab do not always turn out as we intend. For example, this is one sample that was intended to have 20 unit cells, which appear as repeating atoms in each kind of brick that we can see. If this crystal were grown perfectly, we would have boundaries that correspond exactly to these. However, if we zoom in on one smaller region of this film, we can see that one section of the sample is missing this boundary, and thus has a region with essentially double the intended number of atoms. These kinds of defects, however frustrating, are unsurprising, as engineering at the atomic scale is certainly not a trivial task. These defects that occur during growth can completely change the response of the material. Thus, being able to characterize and quantifiably describe a sample is essential to any researcher who wants to understand what material they're actually working with. Previously, this characterization was a menial task, carried out by undergraduate or rather unlucky graduate students who would spend countless hours going through images, counting atom by atom. Now, I want you to put yourself in the shoes of a young, budding undergraduate researcher. You just joined a lab for the first time, and you're ready to hit the ground running with your groundbreaking Nobel Prize-winning research. But then you get this picture, and you're told to count all the atoms in it. Okay, I mean, it looks like a lot, but you can put on a pot of coffee, get a nice magnifying glass, count away. But then you get this picture, and this picture, and this picture, and this picture, and suddenly you're seeing atoms in your sleep. Aside from this just being a tedious and drawn out process, this kind of manual counting has a number of pitfalls. Of course, there will be some human error introduced in the process when you're counting tens of thousands of atoms. But beyond that, this process doesn't give us much insight on the sample and only yields some not very statistically rigorous numbers that give a general overview of the quality of the crystal. To both optimize this long process and generate more statistically sound and useful results, I built a Python analysis platform that is able to identify, measure, and quantify the occurrence of different atomic structures present within these kinds of layered samples. So, how exactly does this program work? Let's start from the beginning of the process. Here we have a STEM image of one of the kind of samples we're interested in. The program I constructed utilizes Fourier Analysis, which essentially means we can isolate and analyze a single frequency within the image. We can generate a fast Fourier transform, or FFT, of this image, which tells us which frequencies and repeating patterns are most prevalent. We can then select one of these frequencies and isolate it in order to analyze the image further. So let's zoom in a little closer. In case you didn't see it right away, I'm zoomed in on a region with a boundary, which is highlighted right here. In this program, we look at the frequency that corresponds to half a unit cell spacing. In real space, this distance is about 2 angstroms. Just for scale, the width of a strand of human hair is on the order of 100,000 angstroms. Throughout most of the sample, we can see this 2 angstrom spacing repeat almost perfectly. However, when we encounter a boundary, which on the macro scale is the end of the repeating structure, we no longer see the same spacing. Instead, we see an increase in the spacing between each horizontal plane by almost half an angstrom. Going back to the full image, we can repeat this kind of frequency analysis and generate a map of the apparent strain in the image. The regions of high positive strain, which are shown in green, correspond exactly to where the boundaries are, because we can effectively think of these regions as being stretched from the normal spacing. However, it's important to keep in mind it doesn't necessarily mean the material itself is experiencing strain, only that we're seeing a different spacing between each horizontal plane. Now we have this image, which is a super easy to see guide as to where the boundaries in the sample are. Even if you could see them before, which isn't always easy by eye, this kind of data is much more binary and simpler for a computer to interpret. That is, we can now take line cuts across this image and analyze each point of that line as either being a boundary or not being a boundary. This is an example of one of these line cuts. We can see these 2 giant spikes in the strain, which are exactly where the boundaries are. Measuring the distance between these 2 peaks in real space, we see that it corresponds to exactly 20 atoms in between each boundary, exactly as we intended. Now, if we repeat these line cuts at every column of atoms in the image, we can generate a map of the unit cell counts that look like this. In the process of making this map, we can also store the number of atoms associated with each line drawn. So in this example, all of these lines drawn correspond to 20 atoms in between each boundary. So you know if we intended to grow a sample with 20 atoms, this region turned out exactly how we expected. However, it's not always that easy. That was just one picture out of the giant stack we had to analyze, and they're not all this straightforward. Here are results from a few other regions of the exact same sample we were just looking at. So even though before it looked like it was a perfect sample, it clearly was not representative of the entire thing. But now we can turn that giant, overwhelming stack of images we were faced with before into 2 distinct kinds of data, quantitative and qualitative. These maps really help us qualitatively understand our samples. We can see regions where growth went well and regions where growth went poorly. But on the larger, more general scale, we can still look at this from a quantitative standpoint. We can actually generate a distribution of the different atomic structures present throughout a series of images, as is shown here. This graph represents over 30,000 atoms counted, an eye-straining process that previously would have taken weeks, now reduced to about an afternoon's worth of running code. Although this code was originally developed with one crystalline structure in mind, the program is actually general enough that it can be applied to a number of layered materials. As the program will be open source, it can be implemented by a wide range of researchers studying all different kinds of layered materials. These pictures represent a currently ongoing application of the code in niobium-3 bromine 8, which is a very exciting 2D magnetic material. However, on a much more fundamental level, this program can ultimately entirely change how we're able to interact on the atomic level with nanoengineered materials. Take this sample for one. Although counting these layers manually would provide some insight on the overall distribution, any interesting patterns or occurrences could very easily be overlooked and lost in a pile of other images to analyze. However, using this program, we can generate a map overlay that clearly highlights the different chemical structures present in this one region of the sample. In particular, we can see this kind of diagonal line that was a result of having the incorrect ratio of elements we were building with during growth. These kinds of layered materials are often grown via molecular beam epitaxy, which means they're grown layer by layer, one at a time. Because of this, if the exact ratio of elements is not correct at each and every layer, we're going to encounter issues like this that we see in the image. However, if we implement this kind of mapping as a part of the feedback loop, anyone who grows these samples will be able to easily identify at which layer things went wrong and can look back into growth conditions and find out what was going on at that time and address it in any future growths. With this kind of analysis readily available for these samples, hopefully we can stop seeing less materials that map like this and start seeing more materials that map like this, bringing us one step closer to bullet trains, 100% renewable energy, and allowing us to connect with others around the world at speeds like never before. Thank you.
Okay, that was amazing, Erin, and what a great start to our conference today. I would now like to open our Q&A round for Erin and ask our audience to type their questions in the chat, or you can privately message me, Daniel, or Neha. And I'm seeing in the Q&A that we have a couple questions already. So Erin, how does having different kinds of structures in a sample change the response of the material?
Yeah, that's a really good question. It's actually very dependent on the material itself. So for example, if you have one kind of perovskite that's a superconductor, but it's mixed with another kind of perovskite that's not, it may stop the superconducting properties, it may not. It really depends on what the exact structures are, how much there is, which is why this kind of mapping is so important, because we don't know exactly how it's always going to affect it one way or the other.
Okay, great. We have another question. When you mapped the counts, why was a lot of the picture empty?
Yeah, it's also a really good question. So I'll go back to that slide actually. So I think everyone can see my cursor. So for example, on this image on the right, we have this really empty region here. And the reason for that was actually to make the data more statistically sound. So we have this issue where if you look at the crystal from one direction, you're going to see these really clear boundaries we saw earlier in the presentation. However, if you just rotate it a little bit, you're not going to see that same boundary. And we can't always make sure we're looking at it from the right direction. So these boundaries we see here are actually a lot weaker than the ones that were mapped. And so just to kind of keep everything as precise as possible, we just don't count these line cuts because we can't accurately determine whether or not this is a true region that we think it would be.
Okay, and our next question for you is, what advantage does your code offer over ImageJ?
Yeah, so over ImageJ, ImageJ wouldn't give you this kind of mapping. So yeah, you can actually use ImageJ to kind of quantitatively look at how much space is in between the things. If you look at, so you can take a line cut of an image on ImageJ and you can get that space, but that's kind of very similar to what you would get from counting it manually, and you wouldn't get this kind of mapping and more visually qualitative results that can be of a lot of use to any growers who want to build samples.
Okay, a couple more for you. Statistically, how is this process better than just manually counting?
Yeah, so as I mentioned before, we now take into account these boundaries that aren't always perfectly clear. But also what we do is we can throw away certain line cuts if we know that we're going to bias the data one way or another. For example, I don't know if I have a picture here that shows it, but if you look at this region, the image here, if you imagine that we don't see a boundary here, right? So we could imagine this is going to be a super long one, but we don't know that for sure because we're limited by the field of view of the image. So if we don't count that and we only count this boundary here, we actually would bias the data towards a lower count, because we're going to count this, let's say this is 20 and this one is 40, we're going to only include the 20, but we can't include the 40 because we don't see it. So this is one thing that you can now take into consideration very easily with this kind of code that wasn't done manually before, along with not counting the regions that have not very clear strain or not.
Okay. Someone's wondering how the pandemic has affected your research experience.
Oh, yeah, that's a really good question, actually. The pandemic entirely changed my research experience. So prior to this, I was doing a very different project that involved characterizing the holders that we used during electron microscopy experiments, the holders for the samples. But obviously, in mid-March last year, I couldn't continue that work as we were all sent home. So I entirely switched my project to a much more computational heavy project. And I think that might be the best thing that's happened in my research experience, because I really found that I love this intersection of programming, material science, and physics. And yeah, definitely it's had a huge impact on my research experience, but it's all been very positive. So one silver lining, I think, of the pandemic.
All right, amazing. I'm not seeing any more questions in the Q&A or the chat. If you do want to ask a question, please do so soon. I'll give it another few seconds. Okay, well, it looks like we are good to move on. I would now like to conclude our Q&A round for Erin. Thank you so much for the wonderful questions from the audience and for graciously answering everything we've thrown at you, Erin.
Awesome. Hi, guys. I'm Neha. I'm a member of the CURBx committee, and I'm excited to introduce our second speaker, Victoria. She's a third-year chemistry and chemical biology major, and she's conducting type 2 diabetes and obesity research at the National Institute of Diabetes and Digestive and Kidney Diseases. So without further ado, take it away, Victoria.
All right. Hello, everyone. Thank you so much for having me. So as Neha said, I conducted research at the National Institutes of Health for 3 consecutive summers, and I'm a junior undergraduate at Cornell. And today I'm going to be talking to you about a protein called Beta-Arrestin 1, which plays a key role in the maintenance of pancreatic beta cells under type 2 diabetic conditions. So just a little bit of background about what my laboratory does. We are trying to combat and address type 2 diabetes. And we want to do so in as natural a way as possible. So emulating how the body normally responds to a high spike in glucose after eating carbs or sugar. And how does the body do this? So it's all down to the pancreas, which is a very vital organ that secretes a hormone called insulin. So after this spike in glucose circulates throughout your body, what happens is there's a counteracting spike in insulin secretion. And the particular cells that are responsible for this are those denoted in blue here. And these are called pancreatic beta cells. And so what happens in diabetes is that there's an impairment in this system where typically these cells, the impairment that we're interested in, is that these cells tend to be very desensitized to high levels of glucose. So even though there's a lot of binding of glucose signals to these cells, there isn't enough of a response of insulin secretion to, counteract the spike in glucose necessary to have glucose homeostasis in the human body for type 2 diabetic patients. And so our lab is focused on how do we actually maximize this insulin secretion despite this impairment in desensitization. And I sort of wanted to zoom in really quickly on a snippet of the membrane of a pancreatic beta cell. And there are 2 proteins that I really wanted to point out that are very important in the millions of types of cascades that happen in this cell. But the real key ones are the GPCR, which is a G protein coupled receptor. And all that means is basically it just binds the glucose signal. It's what actually tells the cell, oh, we have to start reacting now. And then the other protein that comes from that is Beta-Arrestines. And there are different types of Beta-Arrestines, but the one that we're primarily interested in is Beta-Arrestin 1 or BAR1. And the reason that is such an important protein in the cell is because not only does it internalize the signal from the GPCR after it binds glucose, but it also deals with the sensitivity of the cell. So we don't want a cell that overreacts or underreacts to glucose. And in the latter case, this happens in type 2 diabetic patients. And in addition to that, these proteins are very, very involved in the maintenance of the beta cell. So beta cell mass, proliferation, and replication, everything needed to keep the cell healthy. And so we really want to investigate this particular protein and how we can maybe identify targets for it, mechanisms that involve the protein to address type 2 diabetes. And so the next slide that I'm going to show will show just a snippet of the millions of reactions that happen in the cell. But at the end of the day, once we bind glucose, we want to end up reaching the nucleus so that we can, do things with our DNA and then produce the proteins and the molecules necessary to release insulin, which is the final goal. And so there's a lot going on here, but at the end of the day, we're trying to reach this one key master regulator of this ultimate insulin secretion. And that is this protein right here. So this is pancreatic and duodenal homeobox 1 or PDX1. And this protein, as I've said, is a key master regulator. It is the one that is going to really drive this insulin release reaction. And so the goal of our laboratory is to think about what exactly could we maybe target earlier on in the signaling pathway out of the many different proteins and molecules that are cascading downward towards the nucleus, in order to maybe manipulate something earlier on that we can directly correspond to PDX1 and therefore insulin secretion. And we hypothesized that that would be BAR1. And so how did we really examine if this relationship is as important as we think it is? Well, we did some methods in vivo, which involves model organisms, in this case, mice, and in vitro, which really isolates the cells themselves. And we look specifically at pancreatic beta cell lines. And so first I'm going to talk about the in vivo methods. What we quite literally did is knocked down the BAR1 gene in a select group of mice. So we had two groups of mice, both of which were on a high-fat diet for a set number of weeks, but one of them had the absence of BAR1, the other one normal, except for the high-fat diet part. And we did a lot of metabolic tests with that. And what we found, we kind of wanted to do a general overview in vivo of, what impairments are happening here. So first we did a glucose tolerance test. And what we see is that in the control mice, the ones with BAR1 still, we see an appropriate response to, injection of glucose. There's a spike in the blood, and then it sort of decreases over time, which is good. The cells are taking up the glucose, releasing insulin as a result. In the BAR1 knockout mice, what we see is a similar trend, but the issue is that the levels in the blood are still too high. So the cells are still very desensitized and they're not taking up enough. And we now see that this has to do with BAR1. And we also did a glucose-stimulated insulin secretion test. So we just want to see how insulin responds or is secreted in response to glucose signals only, because there are different signals that could release it, but we care about the glucose-stimulated one. And what we see is in the control mice, after this spike in glucose, we see an appropriate response to insulin secretion. That's beautiful. But then in the BAR1 knockout mice, we see this very, very impaired insulin secretion trajectory. Sure, a little bit is being released over time, but it's definitely not at the level needed to maintain glucose homeostasis in the body. And then we also see that we also evaluated sort of the overall health and the maintenance of the pancreatic beta cells, as I'd mentioned before, that BAR1 is responsible for. And we looked at beta cell mass. These markers are a measure of proliferation of the beta cells and replication, and then PDX1, or protein of interest. And we see that in all of these cases, when BAR1 is knocked down in these mice, all of these are significantly impaired. So this already gives a broad overview, and we want to really confirm this particular relationship in vitro, but it's good to see that all these other impairments are happening. It really shows the key role that BAR1 plays. And now we want to investigate this and confirm this in vitro with actual cells. And so we did something a little similar, actually, as to the mice. We had two groups. One group of cells had BAR1, and that's the control siRNA cells. And then the other one didn't. So it was kind of knocked down, degraded. And we just confirmed with a technique called quantitative polymerase chain reaction that BAR1 was indeed knocked down in these cells. And then we stimulated these two groups of cells at an equal but high concentration of glucose for a prolonged period of time. And at each increment of time, we measured PDX1 expression. And we measured it through this qualitative protein identification and amount analysis technique called Western blotting, which is pretty common to use in biological research. And so the results that we found in vitro were actually very interesting. So this graph is the one of most importance, and it comes from quantitatively putting out what we have seen on the Western blot by normalizing these bands with the expression of beta-actin, which is another protein that's always in the cell. So, and what we see is that in the presence of BAR1, so when these cells are normal and they're exposed to these high concentrations of glucose, the PDX1 expression level increases over time, and that's good. The more glucose you have in the cell, the more that the cell needs to work, the more the PDX1 needs to be expressed, and the more insulin that's secreted. That's great. And what's interesting about when BAR1 is knocked down in these cells is that maybe someone would think, well, PDX1 levels would not increase for sure. Maybe they wouldn't even change at all. But what we see is that there's actually a decrease in expression. So as there's more and more glucose in the bloodstream and in the cell, in these cells, we suspect that not in the absence of BAR1, that is what is causing PDX1 expression to be significantly impaired because it's not present there. And so what we conclude from this is that beta-arrestin 1 is a very key modulator of PDX1. And not only do we see this in vitro, we see this in vivo. And so with all these potential confounding variables, we see these in these two types of environments, one isolated one and one that, could have a lot of other reactions be involved, but we don't see that. We still see this direct correspondence. And not only that, but we see this and detect this in a high glucose and high fatty acid or high fat diet environment. And the reason that this is so important is because ultimately type 2 diabetes patients or pre-diabetic patients, they do not have the highest quality of life to begin with. And so when we're thinking about treatments and therapeutics, we don't want to make their lives harder by providing more invasive treatments, right? And so what we just found is a direct correspondence between a protein, beta-arrestin 1, that is near the membrane of a pancreatic beta cell, and another protein that is very deeply embedded in the nucleus of that same cell. So we don't even have to worry about much of the mechanisms or anything that goes on in between, although we would like to explore that, of course. But the point is that when we are thinking about things to target, we don't have to worry about changing the structure of BAR1, the function of BAR1, the mechanism from BAR1 to PDX1. We just have to worry about expression. And this can lead to very effective, very efficient pharmaceutical drugs. And this could lead to the onset of clinical trials within the next few decades. And so that's why this is so promising, is that we have found such a wide range, but direct correspondence between these two really important proteins for the beta cell and to maintain them, to release insulin, and to hopefully improve the quality of life for these type 2 diabetic patients. And so with that, thank you so much for your time. I'm happy to answer any questions.
Thank you, Victoria. That was an amazing presentation, and I'm sure everyone was really fascinated by your research. I know I was. So I'd like to now formally open our Q&A round for Victoria and ask our audience to type their questions in the chat or use the Q&A function. Yeah, or you can always just privately message me, Daniel, or Tara. So, okay, we already have our first question. So, Victoria, how does BAR1 get knocked down?
Oh, yes. So do you mean in the mice or in the cells?
Ooh, they didn't specify.
Okay, so I can talk about both, I guess. So in the mice, it's, I don't know if I should reshare the slide or anything, but what essentially happens is that we sort of combine two types of mice. One that has this sort of dormant protein called CRE, and then the other that has this, that doesn't have that. It has some sequences called LOCK-P sequences, and those sort of enclose the BAR1 gene. Then when you kind of put them in a hybrid, you get this BAR1 knockdown gene, but the CRE protein is what actually kind of cleaves the BAR1 gene. And so it's not activated yet. And so what happens is that you need to inject for a few weeks with something called tamoxifen, or a compound called tamoxifen. And so what we actually did is just for 7 weeks injected tamoxifen into these mice, in one group of the mice. And then we, after that, put them on a high fat diet for 8 weeks. And then for the other one, with knocking down BAR1 for the cells in vitro, it's a process called electroporation. And that basically just means shocking the cells literally, so that they can take up small interfering RNA. And that basically just means it targets the BAR1 gene, degrades it, so that you can't produce any more protein.
Great. Okay, we also have another question. What are the long-term implications of your research?
Yeah, so as I had mentioned before, I think the really key part is that we have found a direct correspondence between a protein in the membrane or near the membrane and another one deeply embedded in the nucleus. And so the implications of that are that you also, I should mention, beta-arrestines are signaling molecules in their own right. And so they don't really fully depend on GPCRs. GPCRs depend a lot on them, but not the other way around. And so what's really important about that is we can really manipulate expression of BAR1 in healthy ways, of course, but in the ways that we want. And it wouldn't affect so much the binding of the signal or anything of that like that. So it seems that this research longer term would have more pharmaceutical implications rather than, for example, something like CRISPR, which is a more like getting in the cell, kind of altering genetics and things like that. It's more something that might bind to BAR1 in a way or get taken up by the cell. And so we're thinking about these types of pharmaceuticals and chemical reactions that could get involved. And the long-term implications are that, someone could just take a pill and that might be enough to really maximize insulin secretion.
Oh, that's great. Okay. We also have one more question. How did you get this opportunity at the NIH to study type 2 diabetes? And what's your advice for getting involved with a similar opportunity?
That's a good question. I think, so I actually had this opportunity since high school. So the summer before my senior year of high school, then the summer after that, then the summer after that. So I did this research over 3 summers. And back then you sort of had you had one big pool of applicants, but nowadays you sort of have high school and now college applicant pools. So it's definitely less cutthroat if you're a high schooler. I don't know if there are any high schoolers in this event right now, but in terms of college, what's really important, I guess, is just knowing that the NIH has sort of a different than usual application process where you kind of just submit your information, like your grades, your resume, CV, your interests in research, a cover letter, etc. And then you're kind of in a database, but you're not really in the program until you have connected personally with a principal investigator from a lab. And then once they kind of put you on a list and then tell the NIH that they want you, then that's how it is. So my advice is just apply as early as possible and reach out to everyone that you're interested in. And I think in terms of like how to do that, it's just like with email correspondence. And the most important thing about that is just to ensure that every email that you send to a principal investigator is very personalized. If you have to copy and paste something, the body of one email to another, then that means it's not specific enough or genuine enough. And a principal investigator can tell when that occurs. So it should come from a genuine place and things like that. Yeah.
That's great advice. Yeah, I'm sure like if there are any high schoolers or college students, they'll definitely appreciate that. We do have one more question. Do you think mRNA delivery could be used to produce the desired protein? And if you continue this project, what next steps would you want to take?
That's really interesting. I've not thought about mRNA delivery because what's interesting about that is that we already have BAR1. So it's more so that we found like a direct. So in changing the expression of BAR1, we've changed the expression of PDX1, and we don't need to change the function of PDX1. PDX1 is the transcription factor that allows for ultimate insulin secretion, and so we wouldn't need to change structure or anything. In terms of mRNA delivery of PDX1, I think that would be very interesting. But in terms of feasibility, I have no clue. I think I don't know, whichever one comes faster would be more efficient. I think with something that targets PDX1, it could be something more in the form of an actual pharmaceutical drug or a compound, but then maybe trying to get right into PDX1 right away would probably require something like mRNA delivery or something more invasive. But yeah, that's a good question.
So those seem to be all of the questions so far. If anyone has any other questions, type them now or forever hold your peace. So I'll give it another 5 seconds. Hopefully that's enough. Okay, looks like there aren't any more questions. So I'd like to now officially conclude our Q&A round for Victoria. Thank you so much to the audience for sending in your questions. And thanks so much, Victoria, for all the wonderful answers and advice. So I'll hand it off to Tara now to introduce our next speaker.
All right, thanks Neha and Victoria. I'm super excited to introduce our third speaker, David Frank. David is a freshman in Dyson from Long Island, New York. He has spent the last 4 years conducting research that combines his passion for sleight of hand magic with behavioral economics. He also recently published this paper in the Journal of Behavioral and Experimental Economics. I'll now hand it off to David to present his research.
Sorry about that. There we go. All right. Hello, everyone. My name is David Frank, and today I'm going to be presenting my research entitled Shattering the Illusion of the Self-Earned Tip, the effect of a restaurant magician on coworkers' tips. And before I begin, as you may have guessed from the title, I am in fact a restaurant magician. I've been doing it now professionally for about 6 years. And I'd actually like to start off this presentation by showing you all a very quick magic trick that I incorporated into my study. So the way I'm going to do this, I have a deck of cards right here. Whoever would like to help me out with this, just put a number 1 in the chat or in the Q&A or whatever you have access to. And Daniel is going to work his Zoom magic and pull you up, and I'll be able to talk to you. So if we could get that, that would be great.
Okay, so we got our first participant. Let me give him privilege to talk. I should have.
Awesome. Hi. Are we good?
Yeah, what's up?
Oh, hey, how you doing? All right. So the other day, well, right before I got on this call, I actually took one card in this deck of cards, turned it face down, put it back in the deck. Now it's on all of you. I want everyone watching right now. Make sure I don't like leave the frame. Keep your eyes on the deck of cards. But the person helped me out right that now I just in the deck of cards, there are red cards and there are black cards. And it's gonna be kind of up to you to figure out what card is face down. So do you think this card is a red card or a black card face down this deck?
Red card.
It's a red card. So I'm gonna switch and go to someone else. Thank you so much for helping me out. Just bring someone else in. We got a red card.
If anyone else wants to participate, just drop a 1 into the chat.
Yeah, that'd be super helpful.
All right, one more person.
Got someone else?
Okay, yep.
Awesome. So within the red cards, we got hearts and there are diamonds. Do you think it's a heart or a diamond?
A heart.
A heart? Awesome. So I was planning on just jumping to someone else for the third part, but I'm just going to stick with you because it seems like not too many people are jumping in right away.
Good. We have more people.
Oh, we got more people, so we'll jump to the next person. So we got a heart. Awesome. Thanks so much for helping me out.
Of course.
Okay.
All right, we're good. Now in the deck cards, there's a whole bunch of values. Got ace to 10 and then jack, queen, and king. What value do you think the card is? The what of hearts?
Queen.
The queen of hearts. That's crazy. Here, check this out. Right here. Make sure I don't go off camera. Nothing like that. Nothing sneaky. Right here in the center of the pack. Right here. We got one card face down right there. You guys said the queen of hearts, right? Because we got a queen of hearts right there. And so I mean, that was just like a quick example of the types of tricks that I would do as I'm going about my duties, performing magic, collecting data for the study. And as a restaurant magician, I go table to table performing tricks just like that. And at the end of my routine at each table, some tables will leave me with a tip, some tables won't. And as I was going through this process, I simultaneously started forming these strong relationships and connections with a lot of my coworkers, the restaurant servers. And after speaking with them, I learned that many of them have to work 2, sometimes 3 jobs, just, put food on the table for their family. And I wanted to see, I mean, that really hit home for me. I wanted to see if I, as the table magician, who was not directly involved in their interactions with their customers, if and how I had any sort of impact on the tips that they brought home. And as I'm sure you're already aware, tipping is an international phenomenon that involves voluntary payments of money as a means of rewarding service providers. And economists will commonly argue that tipping is something known as an efficiency enhancing norm, which is this idea that tipping is the most efficient way to incentivize servers to deliver good service. However, this idea inherently assumes that the tips that servers receive are primarily affected by their own performance and their own work and their own behavior, and not necessarily by extrinsic factors, such as the performance, the work, the efforts of these previously encountered third-party service workers. And there is some research done that shows that these non-tip receiving coworkers can impact customer moods. And some tipping researchers would argue that enhanced moods increase tipping. So in that sense, it's very possible that these non-tip receiving previously encountered third-party service workers, despite not having that direct involvement in the server-customer interaction, it's possible that they do impact the main server's tips. However, these tipping studies that I'm referring to, they tend to focus more on the characteristics of the tipper, a specific service provider, and their interaction, rather than on the effects of these third parties. which is an unfortunate oversight due to the frequency at which we encounter service providers in sequence. For example, at a restaurant, prior to even meeting your server, you may be greeted by the valet, seated by the host, tended to by the busboy. You can order drinks from the bartender, and so on and so forth. So therefore, I wanted to test the effects of a restaurant magician's performance and a restaurant magician's tips on service tips. And to do this, I conducted 2 experiments. The first of which, I performed magic for 270 dining parties at a local family-style Italian restaurant, 6 to 9 p.m. Saturday nights. And prior to arriving at the restaurant, I had a list of, I had a stack of index cards with predetermined table numbers written on them. And when I got to the restaurant, I would randomly assign a table number to either a magic performance or a no magic performance condition. And I simply did this with a coin flip. If it landed on heads, I would do magic for them. If it landed on tails, I would not. Of the tables in the magic performance condition, I then assigned those tables to either a souvenir playing card condition or a no souvenir playing card condition, meaning that I would either leave them with a signed and selected playing card that I just used in the magic routine that I showed them, or I wouldn't leave them with that card as a souvenir. And the way I did this, I would during the trick, I would randomly take out a card from the deck. If that card was from ace to 6, I would leave them with their souvenir card. If it was from 7 to queen, I would not. If it was a king, I would just replace it and select another card and follow the same procedure. I then accepted any tips that they offered me, and I recorded 1, did I do magic or not? 2, if I did magic, I recorded the size of my tip as the magician. And then across all conditions in the study, I recorded the dining party size, server's tip, and the overall bill. And upon analyzing these results, I actually found a treatment effect on group size, such that the size of the tables that received the souvenir playing card were reliably larger than those that did not. And while it's possible this was merely a type 2 error and the result of chance alone, it's also possible that the post-performance order of the playing cards was in some unknown way impacted by the size of the tables that were watching the magic trick. So in order to ensure the internal validity of the study, I repeated this experiment, this time changing the way that I randomized the souvenir, no souvenir conditions, and I simply did it with a coin flip this time. If it was heads, I would leave them with the souvenir card. If it was tails, I would not. As for results, I found that the tables that received the magic performance tipped their servers reliably larger than those tables that did not. However, the size of my tips as a magician had no impact on the server's tips. And here you can see the average percentage tip to the server per no magic condition. And you could see going from the no magic condition, they tipped less than 19% on average, whereas in the magic condition, they tipped just about 21%. And this result is significant for a couple of reasons. Firstly, it indicates that tipping is not fully rational or efficient. As to the efficiency of tipping, that goes back to what I was saying before about the economist's assumption that these third parties don't impact servers' tips. And as for the rationality of tipping, well, the only rational reasons to leave a tip would be either to ensure good future service or to reap some sort of social or psychological benefit of norm compliance. And since tipping can't rationally be expected to enhance the future service of non-recipients of the tip, and since social norms specify only that tips vary with the tip recipient's service efforts, a rational consumer's tipping decisions wouldn't be impacted by these third parties. And secondly, that this result speaks to a debate going on right now within the service industry with regard to tip distribution schemes, or more commonly known as tip pooling. And it provides credence to the idea that, well, if the tips that servers are making are not solely due to their own performance, their own behavior, their own work, but rather the performance and the behavior and the efforts of a series of service workers, well, then these coworkers are contributing to the tips that the servers receive. And then therefore there may be an argument there that therefore they deserve a share of those tips. And then here you can see the total amount tipped to me as the magician per card, no card condition, meaning the souvenir playing card. And you can see that the tables that receive the souvenir playing card tip me as the magician a lot more often than those tables that did not. And the size of my tips were also greater. That's not shown in this graph right here. But yeah, you're going from $123 in the no card condition to over $400 in the card condition. And this significant difference allowed me to establish these sort of big tip to magician, small tip to magician conditions that I then used to analyze how the size of my tip as a magician impacts the service tips. And I ultimately found a null effect of my tips as a magician on the service tips, but this null effect is significant for a couple of reasons. Firstly, it indicates that tipping doesn't engage something known as a resource budgeting process, which is this idea that customers come to the restaurant with X amount of dollars to spend on tipping. And while my research didn't find that this sort of mental tipping budget exists, it is possible that it exists, and my study just didn't find it because it's just so extremely, extremely high, but that's also not very likely. And then secondly, from a more managerial perspective, this result suggests that managers can allow their entertainers and these other third-party workers to accept tips without having to concern over whether they're negatively impacting the tips to their main servers. And in conclusion, servers receive larger tips when a magician performs at their table than when the magician does not. But the magician's tips have no impact on the server's tips. So ultimately, this indicates that the behavior and the performance of third-party service workers can impact the tips to later encountered servers. And this is important for a couple of reasons. Firstly, from a more managerial perspective, if managers know how they can increase the tips that their servers make, they can then actively work to decrease server turnover, which in the long run is going to be very, very beneficial to the restaurant. And then from a more server-minded perspective, well, by knowing the small things that can be done to increase the size of servers' tips, you can then increase their take-home pay, which may be linked to a better quality of life, reduced stress, and greater financial stability. Now, I couldn't finish this presentation without taking a second to thank my mentor, Dr. Michael Lin, for his constant guidance and support throughout this entire process. Here are my references. Thank you.
Thank you so much, David, for presenting your amazing research. I'd now like to open our Q&A round for David and ask our audience to either type their questions in the Q&A box, or you can privately message me, Daniel, or Neha. So for our first question, what would you say are the most significant limitations of this study?
Yeah, so they were definitely, oh, what was that? Oh, sorry, there was like an echo, sorry. Yeah, there were definitely a couple of limitations to the study, including the fact that the studies were conducted by a single magician at a single restaurant, which raises questions about the generalizability of the results. And secondly, the commercial setting of the restaurant didn't necessarily lend itself to the collection of survey data about whether the magic performances truly enhance customer mood. And while I did hypothesize this in the study, I think it would be super interesting to actually get that empirical data to see if that's a true effect.
Great. Someone wants to know how you did the trick, but I'm guessing you're not going to answer that one for us.
Not as fun if I tell you, don't worry.
All right. And our next one is, how did you get interested in performance magic?
Ooh, that's a fun question. Yeah, so I mean, I started doing it when I was about 12 years old. I was honestly on a long car ride back from vacation with my family. I was very bored in the car ride, and I had a deck of cards, started shuffling. And I mean, I feel like everyone as a little kid goes to like that kind of magic phase, and I just never really grew out of it. Got a job pretty early on, and I mean, I've been doing corporate parties and all that stuff, and I love it. Yeah.
Awesome. Do you have any ideas for future directions of research on this topic?
Yeah, so I mean, to preface my answer to this, I should mention a certain aspect of my research that I didn't really touch upon in my presentation. And that is, in addition to looking at the effects of the treatment conditions on service tips, I also looked at how they impact the per person bill size at each table. And I ultimately found that the treatments had no effect on per person sales. However, although the treatments had no impact on per person sales, it's still possible that the overall sales were affected, and my study didn't account for repatronage. So, I mean, it's possible that the magic performances increased repatronage and in turn increase the restaurant sales. So I think it'd be really cool to conduct a future study that examines this repatronage as one of the variables. And also, I think it'd be really cool to repeat this experiment with multiple magicians at multiple restaurants to increase the generalizability of the results. So for example, I think it would be really interesting to maybe do this same experiment at a restaurant in the theater district. Because people might be more in a rush and might not more busy, not as perceptible to seeing a magic trick. Or also maybe at a more high-end restaurant where people might want to focus more on the food, and they may view the magic performance as a sort of distraction from the experience they came to that restaurant to have. So I think those would be two interesting directions for future research, yeah.
Okay, we've got one more, and I think this is going to be our last question for the sake of time. This person makes the maybe unfounded assumption that people tip larger amounts when they experience something novel or exceptional, such as a magic trick or really good service. If this is something that is then generalized to widespread restaurants, would you expect this trend to hold given this exceptional or unique activity would no longer be non-normal?
That's a very interesting question. it would be very interesting to see if this does transition and transfer over to these other service areas and these other service workers. Honestly, I don't know. And I think that would be a worthwhile direction of future research. But honestly, the reason that a lot of studies in the past haven't examined this sort of relationship is that it's difficult to look at this relationship because it would always involve giving certain groups good performance or good service and other groups bad service. So it was relatively well fit for the restaurant performer aspect of this, because I can just not do magic for someone. But it would be a little more difficult to convince a restaurant to purposely give half their customers bad food or something like that.
Okay, well, thank you so much for your great answers to those questions, and thanks to the audience for typing in such insightful questions. And with that, I'm going to conclude our Q&A round for David.
That was great. Okay, next in our amazing lineup, we have our fourth speaker, Narda. Narda is a senior in Cal studying biology and society, and she's interested in health disparities, especially in pertinence to maternal and child health. So I'll now hand it off to Narda to start our presentation.
Thank you. Hello. So I work with the August lab, and my research is on the effects of increased mitochondrial DNA mutations on the macrophage response to Listeria monocytogenes infection. So simply put, how do your immune cells, specifically macrophages with dysfunctional mitochondria, fight bacteria? We all know the mitochondria is the powerhouse of the cell, and it fuels various cellular processes through ATP production in the electron transport chain. DNA in the mitochondria is referred to as mtDNA, and its maintenance and replication is supervised by polymerase gamma, or POG. The mouse model that we have in this experiment has a proofreading deficiency that makes it unable to eliminate these mismatched nucleotides in red. As a result, the mtDNA mutations accumulate, leading to the production of mutated mitochondrial proteins, which will result in mitochondrial dysfunction, such as inadequate energy production. Hence, the mice with the POG mutation, shown here in B, exhibit premature aging phenotypes, such as hair loss, graying, and kyphosis, which is the rounding of the back. These features become apparent as early as 9 months of age in the POG mice. But pictured here is a 13-month-old wild-type mice, and you can clearly see it's not showing these aging phenotypes. Foods such as deli meats and cheeses may contain a pathogen known as Listeria, which causes the disease listeriosis, usually in the elderly and the immunocompromised. Listeria inhabits your liver and the spleen, which will be the focus of our study, as well as the brain and the fetus. This makes pregnant women and newborns 10 times more likely to get listeriosis. Though rare, listeriosis has a very high mortality rate. And currently, there's an active case in New York, Maryland, and Virginia, which has led to the death of one person. And the CDC has traced this outbreak to a type of soft cheese known as queso fresco. Macrophages are the first line of immune defense. Upon infection, macrophages detect and engulf Listeria. In the macrophage, the mitochondria produces MTROS, or reactive oxygen species, which are highly damaging molecules that are thought to aid in killing the pathogen. But for macrophages to clear the pathogens, they first must be activated or polarized. Initially, all the macrophages exist in this M0 non-polarized state, and toxins such as bacterial LPS will induce an inflammatory environment, leading to the activation of M1 macrophages. Afterwards, another cytokine known as IL-4 activates M2 macrophages to resolve the inflammation and repair the damaged tissues caused by M1 activation. And so both of these processes are highly dependent on the mitochondria. So we wonder, what happens if the mitochondria is dysfunctional? Is one phenotype favored over the other? Or is there no activation at all? Thus, this study would provide novel insights into how mitochondrial dysfunction affects macrophage polarization and infectious diseases. Moreover, mitochondrial dysfunction and macrophage activity have been linked to excessive inflammation during old age, otherwise known as inflammation. So findings from this study will contribute to this body of knowledge thereby informing drugs and other interventions for age-related pathologies such as cancer and cardiovascular diseases. So to understand the effects of mtDNA mutations on macrophage response, we address two main questions. One, how does the POG mutation affect the population of macrophages upon Listeria infection? Two, how does the POG mutation affect macrophage polarization? We hypothesize that the mitochondrial dysfunction will inhibit macrophage response in both scenarios due to inadequate energy. To test this hypothesis, we first cultured Listeria and injected the bacteria into the peritoneal cavity of the wild-type mice and the POG mice. 3 days after infection, we sacrificed the mice and we measured the amount of Listeria and macrophages in the spleen and liver of the mice. Our analysis revealed that in the POG spleen had a higher bacterial load than the wild-type mice, which conforms to our hypothesis that the POG mice would be more susceptible to the bacteria. However, in the liver, we saw the opposite, which was really interesting. Though the differences are not statistically significant indicated by the NS, we cannot deny that it may be biologically relevant. So we went on to observe macrophage population in terms of percentage and number. In the spleen, we saw a similar percentage of macrophages in both mild types, but the macrophages in the POG spleen outnumbered that of the wild type. Again, the differences were not statistically relevant, but it raises interesting questions. If there are more macrophages in the POG spleen, why aren't they fighting the bacteria? A similar observation was recorded in the liver, and here, the difference in terms of percentage of the macrophages were actually statistically significant. And so this leads us to our first data suggestion that increased mtDNA mutation is associated with increased macrophage recruitment upon Listeria infection in the liver. As a follow-up, we set out to investigate macrophage polarization. This time, we took cells from the peritoneal cavity of the wild-type mice, the heterozygous POG mice, and the homozygous POG mice, because we wanted to establish a trend of increased mitochondrial DNA mutations. In vitro, we grew the cells in macrophage stimulating media for 7 days, and then we induced polarization into M0 with no cytokine at all. But in M1, we treated it with LPS, and then to M2, we treated it with IL-4. After 48 hours of polarization, this is how the macrophages look like. Generally, the M1 cells seem more rounded, and then the M0 and the M2s were elongated. And then this elongation is mostly observed in the heterozygous POG mice. Then we stain the cells for specific antibodies that are expressed in all peritoneal macrophage cells. And the result shows very similar percent and number of macrophages in both genotypes and phenotypes, which means our experiment worked and we're actually looking at macrophages. Then we went on to stain the cells for CD86, which is an antibody expressed highly in M1 macrophages. In terms of percentage, the graph shows a gradual increment in M1 macrophages as the POG mutation increases. But this trend was not observed in terms of numbers. So this leads us to a more hesitant data suggestions that increased mitochondrial DNA mutations may be associated with increased M1 macrophage activation. Again, we looked at CD206, which is a strong M2 macrophage marker, and all phenotypes express CD206, very much like the baseline I showed earlier. So we took a step further to observe functional markers because CD206 is a surface marker for M2. So we looked at YM1 gene, which is expressed in M2 macrophages, especially when IL-4 is upregulated. YM1 has 304 base pairs, so we know we should be seeing it in this 300 region. And the wild type M2 macrophages showed brighter bands than the het M2, which is indicative of more YM1 DNA in the wild type M2 mice. But then in the homo M2, we did not even see any band at all. And so this brings us to a final suggestion that increased mtDNA mutation may be associated with decreased M2 macrophage response. In a nutshell, the data suggests that the Poggy mice were sicker when infected with Listeria, even though more macrophages responded to the infection. Also, the macrophages in the Poggy mice were more likely to be polarized into M1 than M2, which is like a possible explanation for inflammation associated with aging. But to make more substantiative conclusions, we are going to be repeating this experiment with more mice in order to confirm our findings. And we will be measuring MTROS in order to understand whether the macrophages present are actually capable of fighting the infection. Also, we're going to identify functional M1 markers, such as INOS, and also look at mitochondrial protein expression in the macrophages to help make definite conclusions. Special thanks to the August Lab, and all questions are welcome. Thank you for listening.
That was great. Thank you so much, Narda. I'd like to open up our Q&A round and ask our audience to type their questions in the chat and use the Q&A function, whatever you feel most comfortable with. So, yeah, okay. Looks like we have our first question. Why did you look at spleen and liver in the first part of the experiments, but only at peritoneum for the polarization?
That's a very great question. So for the first part, in terms of population, Listeria has been well studied, and we know that the disease will go into the spleen and the liver. But for the macrophage polarization, that's very novel. We are like the first ones to do that. And so we wanted to use peritoneal macrophages because they are easy to source, and then they're way easier to source than the splenic and the liver macrophages. So yeah.
Great. Okay, we have one more question. What made you interested in studying mitochondrial dysfunction?
Good question. Mitochondrial dysfunction is like a tool I was offered in my lab because my graduate student mentor, she brought mitochondrial dysfunction with her to Cornell. And so, yeah, and I thought it was interesting, especially because it affects aging, and it made it seem like it has societal relevance. That's why I wanted to study it.
That's a great answer. Wow. It doesn't look like we're getting any more questions. If anyone has any questions, please feel free to drop them in now. If there are no more questions, we can conclude our Q&A round. Yeah, thank you so much again to everyone who did send in questions, and thank you so much, Narda, for the wonderful answers. And yeah, I'll hand it off to Tara now.
All right, and with that, we have reached our final speaker for today. Last but definitely not least, we have Hannah. She is a sophomore undergraduate studying archaeology in the College of Arts and Sciences with over 5 years worth of archaeological fieldwork and research experience. I'll now hand it off to Hannah to get started.
So, hi everybody. My name is Hannah Master, and today I will be presenting on wine and getting wasted, the socioeconomic implications of problematized content assumptions in Rhodian and Fourier analyses, which I do recognize is a little bit of a mouthful in terms of the title, but it certainly is all important, at least to me. So let's talk about wine. When we think of the transportation and the consumption of wine, often nowadays we're thinking of a nice glass bottle shipped in maybe a large, lovely wooden crate from Italy or California, maybe even the exciting cardboard box of wine that you can pick up at any hot local establishment. But the question that I wanted to first address was, how was wine transported back in ancient times? Well, it was transported in large ceramic jars like these called amphorae, which just for size-wise kind of comparisons was definitely going to be more like the crate of wine than the bottles, standing around 3 to 4 feet tall and carrying around 25 to 30 liters. Now, this amphora that I've depicted here is a particularly special type of amphora because it comes from the Isle of Rhodes, and particularly from the Hellenistic period, which refers roughly to the time after the death of Alexander the Great in 332 BCE to the beginning of the Roman Empire. But what makes it so special? Well, we know from contemporary textual sources that Rhodes was quite well known for its wine production. So for one example, author Aulus Gellius writes this incredible story about the death of Aristotle in his famous work Attic Nights. And he concludes this section by saying, "Then Aristotle asked for the Rhodian, and tasting it said, 'This is truly a sound and pleasant wine.'" So there's a great rapport here when it comes to Rhodes and its wine production. So how do we study Rhodian amphorae today? Well, practically speaking, when archaeologists excavate different sites around the world, they're never digging up the wine itself, they're digging up the wine jars. And even then, they're usually unearthing fragments of these jars, like the amphora handles that I've shown here. And often what scholars are really trying to study is the wine itself, not the wine jar. But because we can't actually study the wine itself that these jars held, historically, scholars have used Rhodian amphorae as a metric for studying wine trade in the Mediterranean, which makes sense because if you have the wine bottle, you must have the wine, right? Well, it's not usually so simple. Over the course of the history of Rhodian amphora scholarship, scholars have often chosen to add kind of these disclaimers that these jars could have held other things than wine, but then they continue on with the rest of their analyses as though wine was basically the only good transported. And so the first part of my research involved asking a question that's often overlooked in modern scholarship, the question of what else could have these jars held? My goal was to problematize this assumption that these jars were only ever solely used for wine, because that's simply not the case. And so to do so, I drew heavily from publications put out by scholars such as Mark Lawal and Elizabeth Briggs. More specifically, I looked at Mark Lawal's 2000 publication that dealt with contexts in the Athenian Agora, wherein amphorae were thought to have held a variety of different goods. And then I also looked at Elizabeth Briggs's recent 2019 paper that published her finding on some organic residue analyses that she performed, wherein she sampled the interiors of a number of different ancient ceramic jars, Rhodian amphorae included, and then talked about the different goods that they held based off of the organic residue signatures left on the ceramics. So then, with that said, what else could they have held? So goods other than wine have included various cuts of meat, olive oil, fruits like the pomegranate, olives, almonds, olives, pitch, and other objects like beads. And so now as we think about how a large majority of Rhodian amphora scholarship has treated these jars as though they only held wine, we begin to get a sense for why this would be problematic, because we're trying to understand the everyday lives of these ancient peoples. If I were to find one of these jars in a marketplace with butchered meat and just assume that it held foreign wine, then I might have to start making some different assumptions and conclusions about why one would have to have wine in a butcher shop than if I were to simply regard that as any other storage vessel, perhaps having contained imported meat or fish goods. And so then as we're getting into the variety of ways in which these jars could have been used, the next important question to ask is, could they also have been reused? Because as we think about the individual lives that these people led and as they interacted with these containers, it would have been a very different experience if these were meant to be single-use items than if they were more designed to be reused. Think about, say, the different ways in which you would interact with a Chinese takeout box versus something like a mason jar. And so to answer the question, could they have been reused? Pretty simply, the answer is yes. But then the question follows, who would reuse them and why is it important? So let's backtrack for just a second to talk about ancient ceramics. So ancient ceramics are some of the most ubiquitous products that we see in the archaeological record, for the most part because they were cheap to make, easy to replace, and incredibly fragile. So when archaeologists find ceramics, the vast majority of the time we're finding these broken pieces that are essentially ancient trash. They're everywhere because everybody had them. They're much more like the ancient equivalent of single-use plastic and our aforementioned Chinese takeout boxes than they are like more reusable, valuable mason jars. So the next question is, why on earth would these amphorae have been reused? And to answer that question, the second half of my research looked at a case study in the city of Ashkelon, drawing in particular from some work put forth by scholar Kate Burney in 2015 in a presentation that she entitled "To the Dregs, Drawing Meaning from the Rhodian Handles of Hellenistic Ashkelon." So to start off, Ashkelon is a bit of a tale of two neighborhoods. The city of Ashkelon is a port city located on the coast of modern-day Israel and was a prominent port in the Hellenistic period. So the first neighborhood that we'll look at here will be grid 38, which we'll henceforth refer to as the North neighborhood. And for reference, these grids are about 100 by 100 meters in size. The North neighborhood was a pretty affluent one, and we know this because of the types of artifacts that we found there. For example, 60% of all the coins found across the site were located here. The goods they had in this neighborhood had a high degree of variety and diversity. And also in terms of city layout, their technical workspaces were very well separated from some of their domestic spaces like cooking areas or living areas. So in short, this was, as far as ancient standards go, a very nice neighborhood. Now the second neighborhood that we'll look at will be grid 51, which we'll henceforth refer to as the port neighborhood because it was situated next to the port. The Port neighborhood was much less affluent. It was a much less affluent neighborhood than the North neighborhood. And we know this because they didn't have a great deal of coins or jewelry. Their workspaces were in very close proximity to their cooking spaces. So you had technical workspaces overlapping with your more kind of ideally sanitized areas. And also, a lot of the food that they ate was very protein light and would likely have been very easy to get and not very filling. So we found many things like small bird bones that contributed to their diet. So not particularly good meals. So in short, this was a far less well-off neighborhood. So how does this relate to amphorae? Well, the distribution of rhodium amphorae across these two neighborhoods seems to be very unexpected. And it's unexpected because the more affluent neighborhood produced 13% of the rhodium amphorae fragments, and the less affluent neighborhood produced 60% of the fragments. Now keep in mind, the reason why this is highly unexpected is because these jars and their contents would have been considered luxury goods. They had to travel all the way from Rhodes to southern Israel by sea. And so then the next question is, what's going on? Now we started off by talking about wine. Let's talk about waste. So here's a map of the archaeological site that can give us a better sense for where these different neighborhoods were located. As you might be able to tell, this particular map was also used in an educational program by some elementary school students, hence the nice artistic flourish. But so here we see the North neighborhood in all her glory, situated a decent bit inland. And here we see the Port neighborhood, aptly situated right on the coast by the port. Now, when the rhodium amphorae would come into port, they would usually be decanted into smaller vessels for retail and transportation. Keep in mind that these jars are 3 to 4 feet tall and filled to the brim with 25 to 30 liters of whatever good they're carrying. And so they're not going to be particularly nice to schlep inland to the North neighborhood where all the people could afford their foreign products. So then, once that's finished, once the contents have been decanted into these smaller transport vessels, the next question is, where do the leftover jars go? Well, oftentimes they're dumped right nearby the port. They've served their purpose and they're not particularly hard jars to make, so they just go in the trash. And this dump ends up in the port neighborhood. So then, what is it that we're seeing here when we notice these strained proportions of rhodium amphorae fragments across these two neighborhoods? And well, these different proportions reflect that the less affluent neighborhood was reusing these vessels, not for their original luxury wine, but for whatever they could store in them. And what's so critical to notice here is that with this concept of reusing items that were meant to be single use, there's a difference in this practice across socioeconomic lines. The richer North neighborhood gets the luxury import goods, and the poorer Port neighborhood gets the leftover jars. So on wine and getting wasted. The purpose of my talk today has been to illustrate why we need to employ this more thoroughly problematized interpretation of rhodium amphorae and what they could carry. Because it's important for archaeologists when they excavate and study these jars to disassociate these transport jars from their luxury wines, because problematic assumptions like these cloud us from seeing the peoples of the past and how they lived their daily lives more clearly. It's important for us to understand these matters because these people of the past have lived experiences that we in the present want to understand, that we want to preserve, that we want to relate to. And so the story of the people of Ashkelon without a voice, these socioeconomically disenfranchised people of the southern Port neighborhood, it's a story of waste and of the necessity of reuse. And so as we consider how we think about and deal with waste in the present, this case study serves as a historical point of reference where we can see that old familiar story wherein one man's trash truly becomes another man's treasure. Thank you.
Thank you so much for the wonderful presentation, Hannah, and what a great presentation to end our conference with. I would now like to open our final Q&A round for Hannah and ask our audience to either type their questions privately to me, Daniel, or Neha, or stick them in the Q&A box. Okay, someone's wondering, what was the most interesting or your favorite thing that you learned from this?
Oh, that's a great question. I think probably the most interesting thing that I learned from this was that kind of socioeconomic disparity, because I think that even as we're thinking in terms of our own modern experiences, you know, all of us, or for the most part, all of us are college students, so not all of us can afford to have lots of, you know, like nice luxury items that can be reused. And even as we think forward towards how we can live more sustainable lives in terms of, you know, dealing with climate change and issues of waste. One of I think the thing that struck me as most interesting was this idea that we're seeing this even in the past. It's not this new phenomenon of excess waste and of kind of how the necessity of reuse is seen amongst these lower quality items in poorer communities. So I think that to me was kind of the most exciting part of it. The reason kind of why I focused on it in my presentation. But I also just thought that talking about ancient wine in general tends to be a very highlighted topic in academia, particularly in archaeological academia, because it was kind of a very prominent good in the ancient world. And so I thought that it was really interesting to kind of veer off from this academic status quo, if you will, or I'll go with that, to talk more so about the diversity of goods that these people would have been interacting with, because oftentimes when you see data like this, the two questions that you can kind of ask are, okay, well, is it just that the rich people decided that they don't like this luxury wine that they're still continuing to import, and so this is the wine now of the poor people, or is it more so that we're dealing with issues of kind of necessity and survival? So I think that would have to be the most interesting thing that I took away from this.
Great. We have another attendee who submitted a question. They found your presentation very fascinating, and they're wondering, how have you tried to reconcile evidence found in these sites with the construction of the history of ancient peoples when you can't really verify it in a normal sense?
That's a great question. I think that's a question that every archaeological scholar, every historical scholar deals with to some degree, because these are kind of modern interpretations of just the empirical data that we have from the ancient world. And so, of course, there's always going to be this question continually of, when we are interpreting the leftover pieces of the past, how much of what we're interpreting is kind of our own modern biases that we're imposing on the lives of the people of the past, and how much of it is actually the leftover remains of the people who had these lived experiences. And I think that's something that's been kind of incredibly important for myself and for other scholars to keep in the forefront of our minds, is that, first of all, we have historical texts that will often attest to the different ways in which people lived their lives, the different goods that they consumed, which again, like for these less affluent communities, then become few and far between. But we do have texts of how people imagined themselves, how people would like to kind of have been remembered, whether it be through their kind of receipts or their aggrandizing pieces of propaganda that we still have today in modernity. But also then looking kind of towards a more technical side, this movement kind of in modern archaeology to deal with things like organic residue analyses have really increased what we can say about these ancient vessels, because all of a sudden it's not just like, okay, well, this jar could have held olive oil, or it could have held it could have held wine, it could have held fruit or meat, but we can actually sample the interiors and say, okay, well, this organic residue signature is clearly conifer pitch, or we're clearly dealing with some sort of bitumen material, or we have other signatures like that that give us a little bit more insight into the actual like physical goods that they're transporting and interacting with. And so, of course, there is always going to be that kind of scholarly ethical caveat that, like, we don't know what the people of the past experienced, but by the same token, as we progress with the field, we're getting closer, and it doesn't make our research any less significant, because just because we might not be able to tell the exact story doesn't mean that we can't. It doesn't mean that the people of the past aren't owed our commitment to telling it better.
Great, and I think we have time for one more question, which just came in. When you were in Israel, did you find a lot of pottery? And if so, did you ever personally find pieces of amphorae?
Yes. Oh my goodness. So I feel like I definitely would not be like hitting the right audience if I were to say something like, oh, well, if any of you guys have been to Israel and like gone to an archaeological excavation, because that's just, kind of not the right audience. But yes, in short, if anybody has gone to Israel, it's pretty common that you can see ancient pottery on the street. Like, it's not something that you need to go to an archaeological excavation to see. It's not something that you need to even go to a kind of a prominent historical site, because that entire region is kind of a prominent historical site. And so there's been continuous occupation in that region since the Neolithic period. So you can find it on the roadside. A lot of times, actually, archaeological excavations will take place because there's salvage that's going on because construction is happening. And so when you dig down in the ground for, laying the foundation for a new highway or something, you got to bring in the archaeologists to make sure that you're not just tampering or throwing away all of this historical evidence. So yes, in short, pottery everywhere. In terms of rodent amphorae handles, I have found a couple. I have found some rodent amphorae sherds. Again, you're usually not finding, at least I'm not usually finding these 3-4 foot tall jars just kind of hanging out, because just by the nature of the discipline, most of what we're dealing with is broken ancient trash. And so oftentimes if, you know, if the people living back then were kind of around long enough to keep the vessel in shape, then they weren't just throwing it away or leaving it in places where we would likely excavate. It's going to be used until it's broken. And again, they're pretty fragile, so.
Okay, and with that, I think we're going to conclude our Q&A round for Hannah. Thank you once again to our wonderful audience for sending in your questions, and thank you, Hannah, for the wonderful answers. So with that, we've reached the end of our speaker list. I will now hand it off to Daniel to wrap up the conference.
Wow, so that was great. Again, I just want to thank all of our amazing speakers, Aaron, Victoria, David, Narda, and Hannah, for taking the time out of their schedules to share their research that they're passionate about. I'm not sure if you guys have this functionality, but if you can, please click the applause reaction to give them a virtual round of applause. I'm not sure if that's possible, but we'll keep going. We've had a very impressive array of diverse topics, so I'm really, really happy right now. I hope each of you were able to learn something new from all the presentations, as I know I certainly did. If anybody still has any questions, I will drop, or I already did drop the presenters' net IDs below, and I encourage you to directly reach out to them. Also, if you'd like to give me any feedback on how CURBx went, I'd absolutely love to hear it. I included my net ID with the speaker emails. Thank you again for being such a wonderful audience and asking such insightful questions. In a normal semester, you guys would all be eating insomnia cookies and pizza while listening to these presentations. So hopefully we'll be able to get back to an event like that next semester. I appreciate you guys joining us this evening, and have a great rest of your day. Stay tuned for CURBx next semester.